Greetings, dear learners. This is Faculty of Psychology at SRN International School, Jagatpura, Jaipur, Rajasthan. In this video, we are going to discuss the biological basis of behavior, which is a chapter of class 11, subject psychology, and this is the part 2 of this topic. First, here we are going to discuss synapse, a region where communication occurs between two neurons or between an neuron and a target cell, a junction between a presynaptic neuron and postsynaptic neuron. See, presynaptic neuron conducts impulses towards synapse and postsynaptic neuron transmits impulses away from the synapse. Then there are two types of synapses. First is the chemical synapse, such as the neuromuscular junction, and the second one is known as the electrical synapse, that is faster than chemical synapse. This is the diagram for neuron and a synapse. You can see the synaptic gap. The gap is known as synapse. Now we are going to discuss chemical synapse. The first neuron secretes a chemical substance known as neurotransmitter or NT at the synapse. Transmitter acts on receptor proteins in the membrane of the next neuron to excite, inhibit, or modify its sensitivity. Signal is transmitted in one direction. Chemical synapse or events at a chemical synapse. The arrival of nerve impulse opens voltage-gated calcium channels Ca++ influx into presynaptic term. Then we have Ca++ acts as intracellular messenger, stimulating synaptic vesicles to fuse with membrane and release neurotransmitter via exocytosis. Ca++ removed from terminal by mitochondria or calcium pumps. Neurotransmitters diffuses across synaptic cleft and binds to receptor on postsynaptic membrane. Receptor changes shape of ion channel, reopening or opening it and changing membrane potential. See, neurotransmitters is quickly destroyed by enzymes or taken back up by astrocytes or presynaptic membrane. We have to note here that each nerve impulse reaching the presynaptic terminal, about 300 vesicles are emptied into the cleft. Now this is the diagram for excitation and inhibition that I've recently explained here in this video. Now we are going to discuss the electrical synapse. Membranes of presynaptic and postsynaptic cells are fused here and transmission is much more faster. Here it can be, the direction can be bidirectional, and generally it is associated with defensive reflexes. Electrical synapses is characterized by direct channels that conduct electricity from one neuron axon terminal to the next, that is dendrites. Mostly consist of small protein tubular structure gap junctions that allow free movement of ions from the interior of one neuron to the next. Then we have electrical synapses also has gap junctions that allow local current to flow between adjacent cells known as connexons, protein tubes and cell membrane. These are found in cardiac muscle and many types of smooth muscles. The action potential of one cell causes action potential in the next cell, almost as if the tissue were one cell. This is important where contractile activity among a group of cells is. Then we have transmission between neurons, synaptic vesicles, sacs and terminal buttons that release chemicals into synaptic space. Neurotransmitters, which are chemicals released by synaptic vesicles. Here receptor sites are there which are located on receptor neuron for specific neurotransmitters. Now, this is the most important topic of this chapter. We are going to discuss some well-known neurotransmitters. First, we are going to start with acetylcholine, which is released at the neuromuscular junction, which plays an important role in arousal and attention. Loss of acetylcholine producing cells is linked to Alzheimer's disease. Then we have dopamine, which affects neurons associated with voluntary movement, plays a role in learning, memory, and emotions. Loss of dopamine producing cells causes symptoms of Parkinson's disease. Then we have serotonin, which is found throughout the brain, appears to set an emotional tone. Low serotonin levels are implicated in depression. Then we have endorphins, which reduce pain by inhibiting or turning down neurons that transmit pain information. Here also we are going to discuss neuroplasticity because it is important. The brain can be changed both structurally and chemically by experience. Rat studies shows that an enriched environment leads to larger neurons with more connections. Has also been shown in humans. Recent research has uncovered evidence of neurogenesis or the production of new brain cells in the human brain. I hope you have understood everything that I've explained here in this video, and I hope you found it very interesting, and I hope that all the topics are covered regarding this chapter. I thank you so much for watching, everybody. Please like this video, share this video with your friends and family, and put a comment on this video. And please don't forget to subscribe our channel. Thank you.